infrared, and polarized light.
Wow. That's awesome.
Cool.
I was just reading an article today of photography of flowers in ultraviolet and how they have a lot of different patterns that human eyes cannot see, but insects may be able to see and actually help lead them to pollinating them.
That's cool.
And those green dots that you're seeing in the middle of the screen, those are our lasers that give us a scale down here on the ocean floor. And each of those green dots is 10 centimeters apart. So that helps us determine perspective and also size of different things that we're getting close-ups of.
Yeah, I'm kind of content to do the, just do the line again. It's just one little steep bit, a little bit concerned with, but yeah, we can carry on at 0.2 like we were doing.
And again, those brittle stars and shrimps that we're seeing that hang out in the coral and in, around some of those crinoids and things are actually getting sort of lifted up into the current flow so that they can get better access to food and likely are helping clean some of those sponges and corals, keep them clean. And a lot of these species that we've been seeing, like some of the corals, have one specific associate, one shrimp or one brittle star that hangs out and, kind of staked their claim of that.
So Mike, next time we see one of those yellow corals, the smaller ones,
yeah,
we're going to want to take a sample.
Okay. There's a coral coming up, it's a few.
Yep. Might be a winner.
What is your color, coral? I see yellow.
Is this the one?
I'm going to ask Steve to chime in.
That does look like the right one,
Yes.
though.
Okay.
Looks like there's two of them there, so he can
Stop the ship,
pick.
Chris, please.
Bridge now. Request we hold position.
Want to zoom in there, video?
And as we were just discussing, we see that brittle star kind of up on the stalk. Also some brittle stars in the back there scooting along.
This is the one, or are we still figuring it out?
Yeah,
This is the correct species, yes.
All right, full wide.
We're confirming with Steve that he's happy.
Roger.
He wants about half of what you're seeing there.
Yeah, so we're looking for about 20 centimeters or half of the one in front of you.
Okay. The other one is bigger. If you'd prefer that, I can probably set up there as well.
Yeah, that would give it
A little bit more option there. for
Yeah.
getting 20 centimeters.
that seems good.
Okay.
And these corals, though they do kind of have body plans similar to what you might expect to see from a plant, these are colonial animals, and each of those polyps that we're referring to have a little mouth and tentacles, and they're feeding on the debris that's falling through that through the water.
Can we get another zoom before you start sampling since we didn't zoom in
Yep.
on this one? Thanks.
Go ahead.
Come full zoom here, get a nice, really tight.
Very pretty. And so you can really see the polyps in this shot and the tentacles coming off the top of them.
We're good for the zoom, thank you.
Coming wide.
Do we know where we want to put it?
You want to go back for another zoom?
Actually, could we do one more zoom? We have a shore scientist request to see if there are spines guarding the polyps or
Go
not.
ahead, video.
Coming in.
Where would the spines be located on these polyps?
Not sure exactly.
We might be able to get
Okay,
even a
we're
closer
good
look.
for the zoom for now. Thank you.
If we've got it in the coral cutters, we might be able to get an even closer shot for that scientist.
For those of you wondering about the spines, we, according to our shore scientists, coral experts, would see the spines if they were there in a crown around the polyp, and she did not see them when we zoomed in.
Got it.
Thanks, Meredith.
Zoom in there a bit. So you want it down to about where the little guy's hanging on there?
We want about 20 centimeters.
Like this?
Yeah, that should do it.
Not very good. Not cutting very well.
Skeleton seems quite flexible.
I think you might have a distinct piece pulled off.
If I pull, I've still got it like if I pull up now, I might pull the whole thing off. There we go.
Nicely done.
Where is it going?
Full wide, please.
If we could put that, maybe in starboard bio box A, this forward small compartment.
Roger. You follow me around with the arm, Kylie, with the camera.
Gotcha.
And you show me the jaw end of it.
Yep.
Okay. Okay,
Okay.
box out.
If you
Okay.
check out quad view, you can see our sample box.
Let me know if you need me to bump it out more.
How deep can this go in there?
Nice.
Very good.
Very nice.
Nav, can you drop a target? That's number
All right,
063.
close that up.
Sure.
063.
The way the arm was twisting there, I felt like when you're trying to scratch your back in that one tiny spot that you can't reach. But you got it.
Alrighty, I'm going to start us moving again.
Roger.
Bridge, nav. Request we continue tracking our line. Thank you.
Is it getting hotter up here, would you say, or is it the same?
Warm.
In the back row, I think it's about the same, if not slightly cooler.
Okay.
I think it's warmer as I go towards Argus seat,
Yeah,
cooler as I go towards SCF.
I'm comfortable in my seat, so might be up in the front there.
Sorry.
All the engineering sensors look okay. I've got the temperatures pulled up back here.
Okay.
Have you been staying hydrated, Mike?
Me?
Yes.
No, pretty much intentionally dehydrated. Having to sit here for 4 hours. Just in case.
It's not a submarine, you know.
Looks like another one of the.
Zoom in?
Yeah.
I think possibly a big sea cucumber over there.
Yeah.
So this coral, after the previous zoom, the scientists are thinking it's actually
Scooching in.
Plexauridae coral.
What's that blobbish, grayish, dark-colored thing on the top of the screen?
Oh yeah, eagle eyes again.
No, it's not manganese.
Mantis shrimp eyes.
Love it.
Maybe that's your representative animal.
Spirit animal.
Yeah.
Ooh, urchin to the right.
Oh,
Oh, and to the left.
very long spined. Is the one to the left similar to the one to the right?
Yeah, it's coming on screen again at the
Oh, yeah.
bottom.
Some sort of
Wow.
something encrusted on the rock there. For sure.
Yeah, I don't know what that is.
That is an interesting looking rock, if it is a rock.
That urchin's up here somewhere.
That's full zoom.
Roger. Back out a minute, I'll find the urchin.
Yeah, there's two of them that'll pop into view
There you go.
right there and there.
There's your urchin ID, which I'm not going to attempt to
Okay,
pronounce.
come in again.
Coming in.
And a crab or maybe
There's
a
Hey,
shrimp. Sorry.
hey Al, can you read that urchin name
I'm,
for
I'm
us?
looking at it.
It looks like a
Um,
little blue polychaete behind the urchin too.
Yeah.
Aspidodia dematidae. Aspidodia dematidae.
That sounded
That's
good.
my best guess.
You're
That
brave to
sounded
do
good.
that live.
Or I'm silly to do it live. Or both.
There's a very big shrimp.
Right there.
Jumbo.
like
Oh,
a-
wow.
Oh, hello.
Oh, and a little squat lobster. There's so much when we zoom in that we don't see when we're just flying over.
And it looked like there was a blue polychaete near that urchin as well.
Yes.
There's some
Okay, come wide.
long antennae on that shrimp as well.
Yeah,
Oh,
it's reaching
wow.
all the way around the corner.
Full wide.
Another big sea cucumber friend over there as well.
This seems like one of the more biologically diverse little spots we've been across
Yeah,
on this expedition.
abundant. Yeah.
It is one of the shallower.
Yeah,
Mhm.
that's right. Yeah.
Yeah, we're about 1,100 meters between 1100 and
just
1200.
over.
And the geology is great too. We've got 10 for 10 on this one.
Was that your rating or someone who wrote in?
That was me.
I'd agree. It's pretty cool.
Hey Mike, do you see that rock to the left of the lasers right now? It's right next to some critter. Can we get a zoom on that to see if it's loose?
The
Right
one
there.
right now?
Yeah, perfect.
Yep, go ahead, video.
Coming in real quick.
I bet it is.
Looks loose, hey.
Yeah, let's grab it.
All right.
Bridge
Coming
now.
wide.
If possible.
There's a lot of those
Crossby hold
little
position.
squat lobsters all hidden in the cracks.
We're not too laid back, so this should be about where we're going to be.
Yeah, it should be good. In this little cubby hole here.
Hidey hole.
Oh. Anyone else see that?
Yeah.
That was cool.
What was it?
It was the crinoid leaped
Oh,
off
leaped
the
off that
rock
rock?
and
Yeah.
swam like right through Herc's frame.
What's the biology near the rock?
Looks like a crinoid.
Crinoid.
Yeah, unstocked crinoid.
Very
Might
nice.
run away when we grab this.
Where is it? Here.
I can't stop repeating the urchin name in my head now. Aspidodia
It's sounding better.
I know, I'm practicing in my head over here.
I find with scientific names, it's usually third time's the charm.
I would agree with that. You just got to say it out loud a few times to feel it out.
You want to zoom on the rock first?
Sure.
That'd be great.
There, go ahead.
Squat lobster.
All right, we're good.
Coming wide.
It's good there.
Okay.
There's a lot of squat lobsters in this place. I count 3 on the screen alone.
Oh,
Oh.
there it goes.
Oh, it's cool looking.
There it goes.
Well,
They're
maybe
so
it's not gonna
See,
come
I
off.
think
Oh, there it
There it is.
Oh,
lands.
it sampled itself.
They never look like something that's going to be able to move away, and then there they go.
Oh.
See, I said the other day I wanted to see that. I just think that's the coolest way to swim.
Keeper?
Yeah.
Oh,
That
yeah.
was
Okay,
awesome.
full wide.
Where
Would
do
that
we
fit
wanna
one in
put
the
it?
smaller boxes?
Oh,
Yeah.
definitely.
Full
Yeah.
wide. Just hanging out.
Crinoid's a camera hog.
He's like, that's my rock.
Exactly.
Show me the arm. Yeah.
He's really anticipating where it's going to go.
Knows where all the cameras are.
Which one's it going into?
Could we put that in D since it's not going to be floaty?
Roger.
Thank you, thank you.
That's a good Foley.
Very nice. The sampling and
Now,
the Foley.
can you drop a target? That was a 064.
64.
Yep.
And that swimming crinoid that we see can also be referred to as a feather star. And the squat lobsters that we keep talking about, We were discussing this the other day about their name. Did we say that it's because they are squat?
Yes.
Yes.
So they're called squat lobsters because they do kind of look like lobsters, even though they are actually crabs. But if you imagine a lobster that tucked its tail underneath the main part of its body, that's sort of the way a squat lobster looks. So it looks like a sort of compressed, tucked together lobster, but it's actually a crab.
Yeah, and it's related to hermit crabs and mole crabs. So if you take a look at those, you can see the resemblance.
The way I remember is I think of them as very squat lobsters.
Very squat lobsters. They
Lobster.
are. Lobster. Ooh.
It's not even dinner, and I
Oh.
think this has been a really good dive so far.
It's just about dinner, and it's been a really good dive so far. Again, you're going to hear some shuffling in the control van as we step out to eat, as humans need to do. Which again is the reason why we enjoy using our ROVs, or our remote operated vehicles, down on the seafloor, because we can stay down here for long periods of time. And we have our watch teams that cycle out every 4 hours, and we can explore everything that we need to. Chris, have you been relieved yet? Are you still over there?
I'm still here.
I'm getting a question about a coconut incident. Did you hurt yourself while opening a coconut?
Something tells me, is this Justin writing in?
It says, has Aquaman's hand fully recovered from the coconut incident?
I don't know who this is, but I appreciate their care for me. It's on its way.
What did you do? Did you stab yourself with a screwdriver?
No. I had successfully opened the coconut, but in an attempt to get the meat, I had cracked it on the deck, and the shell cut my hand.
That's what
But
you get.
it is okay.
This
I
Z-Virus isn't
patched
terribly
myself
good.
up.
Still, maybe.
What's that?
Aquaman is okay.
Where'd you have it? Doesn't seem to be floating.
All right, if you guys are all set, we're holding position for that sample. I can get us moving again.
Sure. Are you guys still going at 115?
Yeah, 115, tracking a line at point 2.
Roger.
Okay.
Bridge, nav. Request we continue tracking our line.
Maybe it's just
Thank
me,
you.
but I can't get this thing to float at 30 Z bass. Or if it does, it's so subtle. Oh, you know why? I just picked up a rock.
That's a nice looking sponge.
Wonder if it has any critters inside.
I think I see a shrimp.
Want to try to look down inside or not bothered?
Yeah, you can go ahead. Go ahead and zoom.
You tilt down a bit. All right, zoomy zoom. Video, you want to zoom in there?
He's getting situated right now.
Hey, Wayne.
Reach over and press on that zoom button.
Yeah, zoom.
Zoom, zoom, zoom.
Who's home?
Oh, weird.
Looks like the
Somebody
shrimp went down to the bottom. It's hiding.
scared him away. Oh, he's in there. This
Oh,
little white
there's
guy.
I think I saw another
that's
one
cool.
coming towards the bottom as well.
There's one on the top, yeah.
Oh, it's a party in there. And seeing a lot of these little round
That's
things
good,
like
thank you.
to the right as well. I wonder what those are.
Think I'm up above him there, Jess?
What's that?
Am I above him? Oh, just barely.
Your bubble?
Just above the sponge there.
Yeah.
Looks like we've got more of those yellow plexaurids.
Got a fish.
Oh.
We're gonna zoom on the fish.
Yep.
Can zoom a bit more if you want. See if he'll swim into the picture for us.
Give us your best angle, fish.
He got the memo.
Awesome. Looks like a rat tail.
Kenneth
Coming down a little
Sulak,
bit.
do you know which fish this is? If you're still listening in.
There we go.
It's a looker.
There we go. That's full zoom.
Roger. Turn and give you the headshot there.
Hey, thank you.
Okay, coming wide.
Roger. Where are we? 115-ish.
Give me a little bit of a tug there.
Want me to wait?
Is it?
The ship's still adjusting itself on the course a little bit.
Roger. I'll just come back over closer to you. Contact.
Oh. Another one.
I've up the rock there and scared
Coming
it.
up a bit.
I'll figure it out. Yeah, it's definitely warmer in here. That's not good.
Just sliding sideways while we're right for the ship.
Looks like that same white one that we zoomed in on a
Yeah,
little
with
bit
its
ago.
probably polyps retracted.
A lot of coral all of a sudden.
Yeah.
What's just under the lasers now?
That's a big old shrimp.
Okay.
You got some big ones up here.
A closer look.
Sure.
Go for it.
Okay.
Start to feel.
Let me know when you're ready.
Moves.
Yeah, go ahead.
Okay. Oh yeah. Ten more and you got a po' boy. Okay.
Moving on.
Coming by. Bumping.
Now, what are those purple calls?
Appear to be purple.
Yeah, can we zoom in when we get up there?
Sure.
Renee, what do we have, about 200 meters to waypoint 2?
To waypoint 2, yep, 240
Thanks.
from Hercules.
Looks like we're landed there. Go ahead and zoom.
Zooming.
I can
We sampled
center
these
up
the
if
other
you
day,
want.
didn't we?
Yeah, I think this is in the genus Victorgorgia. That characteristic purple in the Lewis Coppercle stars.
Yeah, it's hard to get it
On dive 2.
in the right light to get a good focus. Ah, there we go.
Sorry.
All
I
right.
should be able to full
All
zoom
right,
there.
there we go. Full zoom. And there it is. There it is.
Very nice. Looks like the bottom left is a crinoid occupying one of those precious calls.
Oh, very nice. I didn't even see him. It's nice. Okay, coming wide.
I can get closer if you want to.
No, no,
all
no.
purple screen there.
No, it's okay. That was good. I think they could identify it from what we got.
Ooh, look at that.
Nice.
Hey.
Oh, and a big old fish.
And a fish. You want me to zoom?
Yeah, when ROV's ready.
What do you want to zoom on? The fish?
Actually.
No, the little cluster and on the rock there.
Yeah, go ahead.
The coral.
Okay.
Not sure which one you want to see.
To the left.
To the left.
Yeah.
So when the lasers are on now?
Yeah. Near. That's the one. It's just an anemone.
Zoom in a bit more if you want.
Thank you.
Bumping. Full wide.
Roger. How am I doing, Jess? Do I need more Z bias to?
Sorry?
Do I need more Z bias to make it super sweet
Is that
like
an
you do?
urchin at the bottom? Looking good.
Yeah, just out of frame, looks like it.
Got it.
Come down if you want. That guy there just coming in.
Looks like an urchin. Sounds like an urchin.
That should be good for some zoom.
Okay.
I'm right over the top of you.
Yeah.
Very nice. He's furry. Definitely furry. Coming wide.
Moving on.
Bump. Hey. What's on that stalk?
Another urchin.
Looks like an urchin.
I didn't know urchins climbed stalks like that. Oh wait,
Zoom
or is
in there if you want.
or is it on the ground?
Okay, I'm zooming in.
The stock may have fallen
Zooming
over
in.
first.
No, it's on the stalk.
Yeah, but it's on the ground. Okay.
Right.
Oh, yeah, you're right. It is on the ground. Yep, it's a dead stalk.
How big is that rock that you're right in front of the Hercules when
Sorry.
you zoom out?
Ooh.
Is that too big? It's hard to tell because the lasers are far.
I can tilt down and put the lasers on it.
Yeah, I might want to take it.
There you go.
Oh, yeah.
Yeah, just
20
grab it.
centimeters.
You want me to zoom in on it, see if we want it?
Yeah,
Go,
that'd be
Jess.
great.
Might want to come up a bit there in a sec.
What's that?
Are you thinking about sampling this?
That wall's coming up a bit closer. It's still 20
is.
meters off the deck, but
You want to stop the
That'll
ship?
be
Yeah, are we
rock
sampling
number
this rock here?
We are. I think we are.
4.
Science, are we sampling this rock?
Yeah. Oh, sorry, I can't hear you on the heads up. Yeah, that'd be great.
Ren, you want to
Bridge,
drop a
nav.
target?
That's you.
Hold position.
Okay, as we start to swing,
Yeah, I'll keep an eye
keep
on
an eye on the sonar a little bit there.
Can do.
Take a quick sample then.
Yeah. We have a reference if we need to come back.
Yeah, just in case they had to come off.
Does Wayne know how to work that winch?
Huh?
So does Wayne know how to work the winch?
We'll surely see. I'm gonna take a look over at the nip. real
Oh,
quick.
sorry.
The wrong one.
This lever.
Yeah. Come on.
Why don't you,
Oh, you want to give me some valve?
why don't you come up first on the winch?
Yeah.
Oh, yeah, sorry. Yeah, I could do that.
That's cool, Wayne. I got it.
Come up
Alright.
to the 20-meter ring is blue again, or black.
Yeah.
Before you start, Jess, you want to get
There we
bubble?
go. That should be right.
Get eyes on it with bubble?
It's fine.
Okay, you got it? Okie dokie.
Well, I think we got eyes on it with bubble.
You do now.
All right, you can look over at the rock real quick. Go ahead and push in there, video.
Pushing in.
All right,
right, you guys happy with this free grab here of this rock? Can I go ahead and take it?
Yeah.
You want to tilt up a little bit there?
Sure.
Thank you.
You're going for a twofer again, aren't you?
No way. I learned my lesson.
That's like a pro. You
Very
want to
nice.
give us a
All right, you
QVC
want
spin?
some lasers here first?
Yep.
And a spin of it.
Oh,
would
we got a
be
free
great.
white coal.
All right. Arm's drooping a bit there.
I'm going to do a DVL reset while we're sitting here.
Roger. If you get a chance, come up on that winch a little.
Got a full 360 degrees of iron manganese coating.
So this is probably too large for one of the small boxes, correct? Herc?
Okay.
Probably.
Coming wide?
That is. Yeah, you guys happy with those views there, video or science?
Yeah, we're good on that. Too big for a small bio box, correct?
Yeah, probably, yeah.
Okay. Let's put it in starboard E, then the large forward compartment.
Roger.
Coming
Video
wide.
come wide, please. All right.
Want me to drive bubble?
Please,
Full
and
wide.
thank you. Would you mind looking over a little bit there, too?
I can find the right joystick.
Heh.
No, I keep trying the other
This
one.
is
Obviously does
065,
not
is
work.
that correct, Data?
That is correct, 065.
Ready for salvo?
Yeah, go ahead.
Salvo. Starboard. Say when.
Good. Are there any, oh, sorry. Are there any corals in this box that we need to be aware of?
No.
There's one in A. Okay, so we'll just do a partial out then.
Roger.
Yeah, rock on.
Rock on, girl.
Rock.
That's your third rock today.
I know. I'm being spoiled right now.
That ain't no cobble, that's a rock.
Yeah. Don't you goes.
Bombs away.
Excellent work. Thank you kindly.
Sure.
My pleasure. Sample tray in. Salvo. Normal.
The A team's back, waiting in the back row there.
Hovering back there. Box all the way in, is it? Must be. Okay, I think our work here is done.
Yeah.
That was fun, thank you.
It's fine, guys.
I'll fly anything that's in front of me, yeah. Yeah, you'll want to go at 115 until we get in front of Argus, and then we'll continue our move.
Some kind of coral to the left of the screen. Not sure what it is.
Just do a quick flyby here. I'm pretty far back.
Okay.
No time to stop.
I've identified it as a primnoid. All on
Oh,
my
really?
own. Oh. Steve, where'd you come from?
Zoom in there.
I was just going to say that.
And we all know that is a
Full
fry
zoom.
kid.
Mm-hmm. That's right.
Cool. Okay, full wide. You can move the ship, Chris.
Roger that. Bridge now. Of course we continue tracking our line.
Zoom in there.
All right, those are in the genus Narella. Primnoids in the genus Narella.
Okay, full wide.
Come line.
Man, it is hot up here. Zoom in a bit. Full wide.
That's a pretty stock coinoid.
Go ahead and zoom.
Coming out.
Every time we zoom in, I'm playing how many squat lobsters can I see? And I see 3.
Oh, they're cool from the back as well.
Beautiful.
I'm gonna punch in a little bit here.
Yeah, go ahead.
Let's pull zoom. coming wide.
Now, do crinoids also use the feathery bits to sort of push
All right,
food
come wide.
towards the middle?
That's probably more tube. They're tube feet. They're on the underside. We can't see right now.
Oh, okay.
Yeah. So like
So
other.
closer to the mouth area?
Yeah, like other echinoderms, they use their tube feet to do most of the movement. Those projections are probably just for particle capture.
Got it.
Or
And so
assisted in particle capture.
And then the tube feet reach and grab from the...
Yep.
Cool.
Yeah, they'll use the tube feet to transport down to these grooves on the main part of their arms, and the grooves will then move the kind of condense and concentrate the food particles and move it towards their mouth.
Very cool.
Mm-hmm. Can we zoom in on the yellowfin on top of this rock?
Yep. Second.
That's different from the one we sampled.
Go ahead, video.
So we were having a debate before in the chat about how to identify that particular plexaurid we sampled, and this one is the other, another of that, the other organism we couldn't identify. This is probably Acanthogorgia.
So does
And
this
they
have
have
spikes?
Yeah, so they do have spines, and the truth is you may not be able to see them on the seafloor, but the spines, when they close their polyp, when they retract their tentacles and the polyp's all closed up, you'll be able to see what looks like a crown of thorns, essentially a lot of spiny projections sticking out of the head of the polyp.
Cool.
Yeah. So I'm betting this is Acanthogorgia, and the other one was plexaurid of some type.
Yeah, full wide.
And I think this one was pretty extensively sampled, so I think we should be able to identify it. Looks like something I've seen before.
And again, those green dots that you all see in the middle of the screen are our lasers that give us scale down here. And the two green dots are 10 centimeters apart. And as we're flying through these really interesting geologic areas, I definitely recommend checking out Argus View as well to see the larger context of the areas that Hercules is illuminating.
Zoom in there.
What's that? Yeah, giant guy.
Yeah, that's so that's a very interesting anemone. That was its name is Relicanthus, and it's the only member of its family, I think, and it also lies within a new proposed order of organisms that are related to sea anemones. So actually, technically, it's only related to sea anemones.
Wow.
Yeah. It was
What did you say it was called?
Relicanthus. Relic as in relic. And that was in 2014. So this has been a recent evolution of this.
Okay, full wide, we gotta go.
Yeah.
And another squat lobster. Small one.
Whoa, look at that big crustacean.
But we know that
on top of the screen.
Oh, yeah.
Big crab. We know that that anemone is pretty widespread in the Pacific, so it's been identified off of Central America and South America. It's also been found.
All right.
cracks in these formations are really cool. Well, and thank you to everybody joining us on Nautilus Live right now. Again, we are exploring Jarvis Seamount number 1, which is northwest of Jarvis Island. And we're seeing lots of interesting life and geology down here.
Cool cave there.
Yeah.
Whoa.
Approximately 100 meters left to go to the peak here.
Roger.
There's a batfish over to the 9 o'clock lasers in a little crevice.
Oh, yeah.
6 o'clock.
Yeah.
You can really see how coral density has increased from the dives we were doing previously over the past few days, mostly because we're significantly shallower now. We're, you know, 1,000, 1,100 meters, a depth we haven't seen yet.
We would definitely still call these deep sea corals though, correct?
Oh yeah. Yeah, there's some debate about what deep sea means in the biology world.
Yep.
You know, if you study shallow water corals, deep sea is probably deeper than 50 meters. But if you study deep sea corals, you're probably talking 200 and deeper.
There's that little sliver.
Yeah, and then
unclaimed space.
And then the 50 to 200 meter folks are called mesophotic corals.
Mm.
Everyone's staking out their territory.
Sounds like when people fight about North Jersey, South Jersey, and Central Jersey. Regional humor for you on nautiluslive.org.
Now they think, so there's been some support recently in deep sea biology, fish community world, that there actually may be another zone below the mesophotic called the rarophotic, and that the communities of fishes are actually distinct in that zone. And it seems to be, you know, reasonably well supported. So I'm curious to see if that's the same case in corals.
Yeah, a lot more
lot
here.
right here.